Good morning, children.
Good morning, ma'am.
We started with the new chapter, photosynthesis, and we learned a few experiments in our previous class. And I want you to summarize at least a few experiments. From memory, I know you might not have learned yesterday, but still from your memory power, you can just recollect and say what you had learned in our previous class. I want you to explain the experiment conducted by Priestley. Who will say? If I ask who will say, nobody will be there. Naureen, I'll help you. Just start the experiment.
He observed that a candle burning in a closed space extinguished, and a mouse would suffocate in a closed space. And then he kept a mint plant inside it, and the candle continued to burn, and the mouse stayed alive.
What was the reason? What did he conclude from that?
The plants restored the whatever was removed by the burning candle and the breathing animal, that is carbon dioxide.
And Jan Ingenhousz, he also repeated the same experiments in both darkness and also in presence of light, and he tried out with aquatic plants. So what was his observations? Steffi, what did he observe upon carrying out the same experiment using an aquatic plant?
He found out that in the dark there was no oxygen. Oxygen bubbles weren't formed, but in the light, oxygen bubbles were formed.
Okay, indicating that photosynthesis can take place only in presence of light. So let us continue. Julius von Sachs, so another scientist who carried out different experiments related to photosynthesis, and he proved that glucose is produced when plants grow. When plants grow, it indicates that photosynthesis is also taking place, and glucose is produced when plants grow, and it is usually stored as starch. You know that it is during photosynthesis, the growing green plants, during photosynthesis, they will produce glucose, and this glucose will be stored in the form of starch. So he was the one who proved the production of, so about oxygen release, you already learned about the experiments carried out by two scientists which proved that oxygen is being released by the plants when they carry out photosynthesis, but glucose is also produced and it is stored in the form of starch. These findings were carried out by Julius von Sachs, or his findings proved that glucose is produced when plants grow and it will be stored in the form of starch. And he also conducted studies later to show that chlorophyll is located in special bodies known as chloroplast. From 8th standard, you are learning about chloroplast. This year also you learn chloroplast is a type of plastid. And chloroplast is a cell organelle which contains chlorophyll. The green pigment chlorophyll is present in chloroplast, and this organelle is found only in green plants. And chlorophyll can be also found in certain green colored organisms also, like blue green algae. So there the photosynthetic chlorophyll is present. But other than that, generally in plants, plastids are present, and chlorophyll is located in specialized bodies known as chloroplast present within plant cells. And he also found out that, or his findings proved that it is in the green parts of the plant that glucose is being prepared. So what were the findings of the scientist? Many points he proved. One is that when plants are growing, they will produce glucose, and this glucose will be stored in different parts of the plant as starch. And later, further studies he conducted, and he found out that in plant cells, the pigment chlorophyll is present in some specialized organs or organelles known as, or special bodies known as chloroplast. And it is only in the green parts of the plants, wherever this chloroplast or wherever these pigments are present, there only photosynthesis took place. So these are the findings of the scientist Julius von Sachs. Next is about the scientist T.W Engelmann, and from 1843 to 1909. So let us see what were his findings, or what were the experiments conducted by this scientist. He split the light using a prism. So splitting light using a prism, I think in class 9 or 8 you are learning in physics. So splitting light using a prism. So what he did was he split the light using prism into its spectral components. So when light is passed through a prism, it will split into its components, into different components of light, and then illuminated. So using those components. So once light was split into its spectral components, the visible spectrum of light was split into different components, and using that he illuminated, or he lighted green alga, cladophora. The name of the alga is cladophora, and this cladophora is an aquatic plant, it's an alga. It was placed in a suspension of aerobic bacteria. So these plants were kept in a suspension containing aerobic bacteria with an intention. We will learn why aerobic bacteria was added to this medium. I'll repeat again. What he did was he split light into its spectral components using a prism, and then with the help, or with this light, with these components of light, he illuminated, he lighted up because for carrying out photosynthesis light is needed. So this light was provided to cladophora or alga, which was growing in a suspension of containing aerobic bacteria. Hope you know about aerobic and anaerobic bacteria. Aerobic bacteria, they can survive only in presence of oxygen, whereas anaerobic bacteria, they don't require oxygen. Sometimes it can even poison. Presence of oxygen can even poison these anaerobic bacteria. The bacteria were used to detect the sites of oxygen evolution. So if this plant is there, plant is growing in the medium where these bacteria are there, being aerobic bacteria, they need oxygen to survive. So they will be found in regions where the oxygen is getting evolved. Where oxygen is available, there these bacteria will form colonies. So just to spot out the areas where, or regions where oxygen was evolved, these bacteria were included in the medium. He observed that bacteria accumulated mainly in the region of blue and red light. So you know that the light, when it is split, that VIBGYOR, so you can see so many components of light. So wherever, or in the blue and red light of the spectrum, split spectrum, there the bacteria accumulated. So in the beginning, using a prism, he had split the light into different components. Using that, he illuminated the plant. So wherever blue and red light was there, there the bacteria accumulated, which indicated that photosynthesis is happening more in these areas where the plant is lighted up with blue and red light. The first action spectrum of photosynthesis was thus described. So action spectrum is it's a graphical representation to show any physiological activity at different wavelength of light. So wavelength of light can be plotted on one side, and any physiological activity on the other side. So here they could notice that wherever blue and each light is having a fixed wavelength. So wherever blue and red light was there, in those regions the photosynthesis was maximum. So action spectrum is just used to denote or to represent any physiological activity at different wavelengths of light. So this is how, and here with the help of the scientists, the first action spectrum of photosynthesis was described. It resembles roughly the absorption spectra of chlorophyll A and B. If you compare with the action spectrum of photosynthesis, you can find that it was resembling the absorption spectra. So the difference between absorption and action spectra is action spectrum will show the physiological activity. So here it is photosynthesis. Photosynthesis at different wavelengths of light. Whereas absorption spectrum will indicate the absorption of light at different wavelength. How much light is absorbed at a particular. So here it is chlorophyll A and B. So in chlorophyll there are different pigments, main pigment, accessory pigments, different pigments are there. So their absorption spectrum, so how much chlorophyll A will absorb, or what is the amount of light, or is amount of the quantity of light absorbed by chlorophyll A and B. So absorption will be indicated by absorption spectra, and the activity taking place at different wavelength is expressed with the help of absorption action spectrum. So it is more or less similar to the action spectrum of photosynthesis will resemble the absorption spectra is plural, spectra of chlorophyll A and B. That also indicates, so when chlorophyll is absorbing more amount of sunlight, there the photosynthesis will be maximum. So that was very matching with the absorption spectrum. Okay, wherever that means the chlorophyll absorbs, chlorophyll A and B, they will absorb this light, and there the photosynthesis rate will be maximum. And in red and blue light, in different wavelength, I said no, in red and blue light, it was found to be maximum. The maximum physiological activity were found in those wavelength, in blue and red light. So this was matching very closely. By the middle of the 19th century, it was discovered by the middle of the 19th century, it was discovered that plants use light energy to make carbohydrates from carbon dioxide and water. So from or by the middle of the 19th century, so with the help of the scientists, it was proved that plants use light energy, or plants use carbon dioxide and water, and they will prepare carbohydrates using carbon dioxide and what I'll repeat once again. I got distracted in between. So by the middle of the 19th century, it was proved, scientists proved that photosynthesis will be carried out by the plants. That means plants make carbohydrates in presence of sunlight using carbon dioxide and water. So that was proved clearly with the help of the experiments carried out by different scientists. The empirical equation, arrow has a little gone up, don't mind it. Empirical equation means the basic form of the equation of photosynthesis, CO2, H2O, means the basic form. In the basic form, it's not a balanced equation. The basic form they have given, carbon dioxide and water are the raw materials, and we get CH2O. CH2O represent the carbohydrate. So we can add enough number of atoms to that. But this is the basic form, CH2O plus oxygen. So that is the empirical equation for the process of photosynthesis, where CH2O represents a carbohydrate. It can be any carbohydrate. Like here in photosynthesis, it is glucose. And later it will be stored in the form of another carbohydrate, that is starch. So once again, I'll repeat this experiment. You want me to repeat? Or you have
Yes, ma'am.
You will explain? No.
Repeat, please.
So T.W. Engelmann, he was the one who used light, or who used a prism to split light into its components, and using these different components, he illuminated the different components of the light comprising of or consisting of different wavelength was focused on, or this plant was illuminated with light of different wavelength received or obtained by splitting white light using a prism. And the plant, the specimen he used was cladophora. In this cladophora, it's an aquatic plant. It was placed, or it was grown in a medium containing aerobic bacteria. Aerobic bacteria were used to spot out the regions where the oxygen is getting evolved, which indicates that once oxygen is evolved from a particular place, you can understand that there the photosynthesis taking place. So in order to spot out those regions, these bacteria were added to the medium. And he found that it was in the blue and red light. So wherever red and blue light was falling on the leaf of this cladophora, there the bacteria accumulated, indicating that there the oxygen evolution is more, which means there the photosynthesis is maximum. So that means photosynthesis is not taking place at the same rate in different wavelength. There are certain wavelengths where it will be maximum, where the photosynthetic rate will be maximum. This was the first action spectrum. Action spectrum, it is a graphical representation of physiological activity. Here it is photosynthesis at a different wavelength of light. And he described the first action spectrum of photosynthesis, and this was very closely matching with the absorption spectrum of chlorophyll A and B. So chlorophyll need to absorb solar energy. Only then photosynthesis can take place. They need to trap the solar energy. So this graph was matching with the absorption spectrum of chlorophyll A and B. So the chlorophyll maximum absorption by these pigments took place in red and blue light. The wavelength of red and blue light, there the maximum absorption of sunlight took place. That also indicates that it is in these regions that the photosynthesis is taking place at its maximum. And by the middle of the 19th century, with the combined effort of different scientists, it was proved that plants use light energy to make carbohydrates utilizing carbon dioxide and water. And the empirical equation is like this: carbon dioxide plus water, or CO2 plus H2. It's not a balanced equation, just the representation, the basic form. CH2O. CH2O indicates a carbohydrate, can be here in this case it is glucose, plus O2. Cornelius van Niel. He is the next scientist. He was a microbiologist, and he conducted studies on purple and green colored bacteria. So depending upon the pigment present, purple and green bacteria, he conducted studies on these particular bacteria. And he demonstrated that photosynthesis is a light dependent reaction in which hydrogen, so anyway you are getting, like, glucose is being prepared, apart from glucose as a byproduct, oxygen is being released. So the next attempt of the scientist was to find out from where this oxygen was released. The two main raw materials used were carbon dioxide and water. Is it both contain oxygen? Carbon dioxide as well as water contain oxygen. So they wanted to know whether this oxygen released is from carbon dioxide or from water. He demonstrated that photosynthesis is a light dependent reaction, because photosynthesis needs light to take place. In the absence of light, it cannot happen, in which hydrogen from a suitable oxidizable compound reduces carbon dioxide to carbohydrates. So hydrogen from an oxidizable compound. See, carbon dioxide has to get reduced to carbohydrates. It is the hydrogen from an oxidizable compound that will convert it into carbon dioxide into carbohydrates. So this is expressed as 2H2A. Okay, why it is written A is it cannot be water in all the organisms. So in some cases it can be hydrogen sulfide. In purple and green bacteria, instead of water, it is hydrogen sulfide. So that is why the alphabet A is given instead of writing O. It can be O or it can be S depending upon the organism. So 2H2A plus carbon dioxide, we get 2A plus CH2O plus H2O. Okay. See the same equation. The first part, this indicates, in the previous equation we represented it as water, H2O, then carbon dioxide. Okay. And this is the carbohydrate, water, and oxygen is also released. So this from where that we want to know, this A got released from where. But from this equation you can make out, once again I'll repeat, Van Niel, he conducted studies using purple and green bacteria, and he demonstrated that when photosynthesis, the light-dependent reaction, when it takes place, some oxidizable compound is releasing its hydrogen to convert carbon dioxide into carbohydrates, and this is represented as 2H2A plus CO2 gives 2A plus CH2O plus H2O. We'll come back to this. In green plants, water is the hydrogen donor. So I told you this hydrogen is necessary for converting carbon. Carbon dioxide alone cannot form carbohydrates. Carbohydrates itself means these are compounds consisting of carbon, hydrogen, oxygen. So carbon and oxygen is already present in carbon dioxide. Only if hydrogen also combines with that, it can become a carbohydrate. From where this hydrogen was given. Once hydrogen is given, the remaining oxygen molecule is also released as a byproduct. So in green plants, water is the hydrogen donor. And once hydrogen is donated from water, it will get oxidized to form oxygen. So in plants, in green plants, the oxygen is released from water. Whereas in purple and green sulfur bacteria, their hydrogen sulfide is one of the raw material instead of water. Hydrogen sulfide is the hydrogen donor. As a result, oxygen was not produced as a byproduct when these bacteria carry out photosynthesis. Instead, what they produced is sulfur or sulfate. Compounds of sulfur or sulfate they produced instead of oxygen. This clearly proves that it was from water, or it was from in green plants, it was from water that oxygen is released. So when he conducted experiments using these bacteria, we could understand, it was proved that it was not from carbon dioxide but from water that oxygen was released. Because when he conducted the same experiment using purple and green bacteria, which are not using, these bacteria do not use water, instead they are using hydrogen sulfide as the raw material. If carbon dioxide was releasing oxygen, then these bacteria, as a result of photosynthesis taking place in these bacteria, oxygen should be released if it was from carbon dioxide. Since it was from hydrogen or hydrogen sulfide, so they could prove that hydrogen sulfide, being hydrogen sulfide is one of the raw material, instead of oxygen they could get only sulfur or sulfate. If it was carbon dioxide, because carbon dioxide is common in both plants as well as common raw material in plants and bacteria. If oxygen was released from carbon dioxide, definitely we could get oxygen from the photosynthesis of these bacteria also, but we did not get. Instead we get sulfate, which clearly proved that it was from water that oxygen was released. And water also, when water is donating the hydrogen atoms for reducing carbon dioxide to carbohydrate, it will get or it will get converted to oxygen. Once hydrogen is gone, it will change into oxygen, and that is released as a byproduct. Have you understood how did they prove that oxygen evolved by green plants is coming out from water and not from carbon dioxide? So when it was carried out only in plants, they were not able to prove whether it is from carbon dioxide or from water. So the same experiment using photosynthetic bacteria was carried out, who doesn't use water as the raw material. Instead, they are using hydrogen sulfide. For these bacteria, hydrogen sulfide is the raw material. So when the experiment was carried out, the expected byproduct was not released. So normally in photosynthesis, oxygen is the byproduct. But when this was carried out in bacteria, oxygen was not released. Instead, what was released? Sulfate or sulfur was released. So from that, it was very clear that it was not from carbon dioxide, but from water that oxygen is released in the case of green plants. Any doubt? Did you understand? Did I confuse you?
Could you say that once
Same.
more about
Ah,
this
Yes, Gayatri, I'm waiting. Speak.
Yes, ma'am, this one about the production of
Ah.
oxygen from
You want me to explain?
Yes, ma'am.
Okay. So what was the intention? Did you understand? What did we or what they wanted to find out? You know that when photosynthesis is taking place, what is the product form, children? What is the product form? What is the product form when photosynthesis takes place? Simple question, simple answer. Tell me what is the product form when photosynthesis takes place. Gayatri, tell me.
Oxygen.
Yes?
Oxygen.
Oxygen is the main product?
No, ma'am. Glucose.
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 And oxygen is the byproduct. Okay? And the raw materials used are water, H2O, and carbon dioxide, CO2. Both carbon dioxide and water are having oxygen in it. Oxygen atoms are present in it. Am I right? And scientists wanted to find out whether it is from carbon dioxide or from water that the oxygen was evolved as a byproduct. Goal or the aim, did you understand what they wanted to find out, Gayatri? Okay, so when it was carried out in plants, they thought it could be from carbon dioxide or from water. But when they carried out a similar experiment, they carried out photosynthesis, or they observed the photosynthesis process taking place in purple and green bacteria. What is special or how they are different from plants? In plants, the raw materials are water and carbon dioxide. But in these bacteria, the raw materials for photosynthesis are hydrogen sulfide, not water, hydrogen sulfide and carbon dioxide. Okay. And as a result of the photosynthesis taking place in bacteria, the carbohydrate is the main product that was produced. Along with the carbohydrate, what was the expected product? Tell me, what is the expected product along with carbohydrate?
Oxygen.
An answer. Yes? Yes, Nandana?
Oxygen.
Oxygen, just like in any other plants, they expected oxygen to be the product, byproduct. But did they get it? No. Instead, what was produced? Sulfur or sulfate. Okay? Sulfur or sulfate was formed instead of oxygen. If carbon dioxide, see, in both green plants and in bacteria, the common raw material was carbon dioxide. And if oxygen was released from carbon dioxide, when we carried out the experiment in these bacteria, we should get oxygen because that was common for both. But in plants, we got oxygen, and in bacteria, we got hydrogen sulfide. So the difference was only in, sorry, not hydrogen sulfide, we got sulfur or sulfate. So the difference was only in water, whether it was water or hydrogen sulfide, that made the difference. So from there, it was clear that oxygen was released from water. If it was not from water, but if it was from carbon dioxide, in purple bacteria also we should get oxygen. But we did not get oxygen. Instead, we got sulfur or sulfate. Carbon dioxide was common in both, but what was different? Hydrogen sulfide and water. That made the difference. So from that, scientists understood that it was from water that oxygen was released. Clear, Gayatri?
Yes, ma'am. Thank you.
Yes?
Yes, ma'am. Yes.
Very
It's
clear.
clear.
Some
What's ma'am?
very clear.
It's very clear, ma'am.
So we'll continue with the same slide. Thus, he inferred that oxygen evolved by the green plant comes from H2O, water, and not from carbon dioxide. This was later proved. So further confirmation of the experiment was carried out with the help of radioisotopic techniques. About isotopes you might be knowing. Radioisotopes means radioactively labeled isotopic techniques. You will learn about that in higher classes. So the same findings were confirmed, were again proved by using radioisotopic techniques. So the overall equation, now we have got a proper equation for photosynthesis. 6CO2 plus 12H2O gives C6H12O6 plus 6H2O plus O2. And we can even write this equation like changing 12H2O to 6H2O. Then in the products, you don't have to mention water. Both are correct in 10th standard textbook, both are given. Did you understand what I said? Here 6CO2 plus 6H2O C6H12O6 plus O2. Okay. Clear, children? No response. Okay. In any ways, you can write, if you're writing 12 here to make the equation balance, here 6H2O should come. If it is 6 here, you don't have to mention water over there. And all those who haven't sent me the project, please do send it today itself. Okay. So hope you have understood. Doubts are cleared. And what is the equation for photosynthesis? Rijas?
6CO2 plus 12H2O gives
C6.
C6H12O6 plus 6H2O plus 6O2.
Okay. And we learned about the experiments of two scientists. And please summarize the experiments of those two scientists. The first one we learned about Punya. About which scientist we discussed first?
Von Sachs.
Tell me, very briefly, you can tell me the experiment carried out by him.
He proved that glucose is produced when plants grow, and it is stored in the form of starch. And then chlorophyll is present in specialized bodies called chloroplasts.
Okay. Vidur, next one, next scientist.
Next is an Engelmann, T.W.
What? Repeat once again.
Well, I don't know the pronounce the scientist name. T.W. Engelmann.
Pronunciation, Malavika.
T.W Engelmann.
T.W Engelmann. Continue.
He split the light using prism
News reading is going on?
uh,
In between looking here, then facing the audience. Okay. You say as you have understood.
into spectral command, and he used bacteria to detect.
He used?
Bacteria to detect oxygen evolution.
What bacteria he used? What type of bacteria? Aerobic bacteria. Why aerobic bacteria was used? Because they will accumulate near the regions where photosynthesis is maximum, where oxygen evolution is maximum. And just tell me in which areas the photosynthetic rate was maximum?
In blue and red.
In blue and red color light, wherever this red and blue light was falling on the plants, there the photosynthesis was maximum. Okay. Thank you, children. See you next class. Please do read.